السلام عليكم ورحمة الله وبركاته. موضوعنا اليوم اللي هو الميديكال فيسيولوجي، المحاضرة الأولى اللي هي Physiology of Nerves and Muscles. طلبتنا الأعزاء، هذه المحاضرة تأتي في الكورس الأول لكلية الصيدلة، المرحلة الثانية، جامعة جابر بن حيان الطبية. محاور محاضرتنا لهذا اليوم هو Structure of Neuron, Function of Neuron, EXCITATION, CONDUCTION، واللي راح ناخذه به هو الأكشن بوتنشيال. والمحور الثالث اللي هو الساينابس. المحور الرابع اللي هو المسلز، ناخذ السكيليتال مسلز، سموث مسلز and cardiac muscles. طبعا اللي يهمنا بالمسلز هو what mechanism by which contraction and relaxation taking place. Physiological of nerve and muscles, cells of the nervous system. Nervous tissue consists of two principal types of cell. Number one, neurons, and number two, glial cells, which is supporting for a neuron. المحور الأول اللي راح ناخذه اللي هو structural of neurons consist of cell body, dendrites and axon, myelin sheath cells, nodes of Ranvier and the later axon terminal bundle. The structural of a neuron, this is cell body. Another figure that shows the structure of a neuron. This question is very important on conduction velocity in a neuron. This is a question in this structure. In this lecture, we need to give the answer for this question. Notes: axon may be insulated with myelin with increased conduction velocity. Breaks in the myelin sheath occur at the node of Ranvier. طلبتنا الأعزاء، يعني at the site of node of Ranvier there is no myelin sheath. It's very important. Later we explain this notes. Another shape of and size of a neuron, there is bipolar neurons, unipolar neurons and multipolar. This is bipolar, this is multipolar neuron, and this is unipolar neurons. Another cells is supporting for the neurons is glial cells. Glial cells give to myelin and provide the support and protective of neurons. The glial cells divided into two types according to the site or position. In the central nervous system there is oligodendrocytes which act as the myelin sheath. Astrocytes act as for providing neurons lactate and maintain stable potassium in the brain extracellular fluid and synthesizing neurotransmitter precursor for neuron. Ependymal cells lining with the cerebral vertex and another microglia. These highly reactives act as the immune system as cause them to proliferate and become phagocytic processes. In the peripheral nervous system, the glial cells consist or include the Schwann cells, supporting cells. This give the myelin sheath for axon in peripheral nervous system, and another satellite cells surrounding the cell bodies and have many same function of astrocytes in the central nervous system as we show previously. From this figure, it's very important to take the face mask against the COVID-19. This is a break in this lecture to prevent the COVID-19. This is picture is the true and another picture is false and false. The topics, another topics, the second topics in this lecture is function of a neuron, excitation, conduction is very important, the action potential. When we experiment in a neuron, we give to minus 70 millivolt. This is indicated to that this of the resting membrane potential is present in the body cells. To explain that, we explain the factor that contribute to membrane potential. Two ions are responsible for this action potential is sodium and potassium. The entering of sodium and potassium, this is depending on sodium potassium bond. This is a protein present in cell membrane act as the bombing of the sodium out the cells and entering the potassium within the cell. Another ions that give the resting membrane potential is the chloride phosphate protein. The phosphate and protein in the intracellular is more than the extracellular, and from this table we give the negative charge within the cell and positive charge outer the cells. This is in a part of this figure give to polarization of cells. From that we must be explanation the action potential. Action potential is very rapid changes in membrane potential that occur when nerve cell membrane is stimulated. Resting potential in a neuron is equal to typically minus 70 millivolt, and then after depolarization reach to positive 30 millivolt. Is very short period, just a few milliseconds. This is figure consists of the action potential. This the resting potential minus 70 millivolt when depolarization reach to positive 30 millivolt, and then repolarization reach to return to the resting potential. The resting membrane potential different from cell to the another cell. Nerve cell minus 70 millivolt, skeletal muscle minus 90 millivolt, and cardiac muscle 85 millivolt, and the small muscle nearly to negative 50 millivolt. And the action potential is very important, and there is many stages that occur in action potential. Action potential, the first stage is depolarization. Depolarization occurs after the sodium channel completely opening, and then the sodium enter to the cells. The action potential is very important when they reach to threshold stimulus. But when the action potential not occur because of there is very less stimulus, not reach to threshold stimulus, and there is no action potential. The threshold potential generally from 5 to. This is the stage of the action potential: resting point, resting stage, and depolarization stage, and then repolarization stage. So from that stages of action potential, resting stage, this is the resting membrane potential. Before the action potential, the membrane is assayed to polarized, and this is the responsible for resting stage is the sodium potassium channel. In depolarization stage, there is a sodium ion channel are opening, and then the sodium enter to the cell and is reach to depolarization stage. After that repolarization stage, the sodium channel is blocked and the potassium channel opening more than the normal. The rapid diffusion of potassium ion into exterior re-established the normal negative resting membrane potential. This is called repolarization. So the repolarization stage is depending on potassium, rapid diffusion of potassium. Another role of another ion is very important, such as protein, phosphate, sulfate. This is give the negative charge within the cell. But another ion is very important. This is calcium. Calcium is very important depending on voltage gated calcium channel. Calcium is extracellular ion more than 10,000 times greater than extracellular than the intracellular. Calcium channel, sodium channel is slightly permeable to sodium ion, but the calcium is about 1000-fold greater than of the sodium under the normal physiological condition. So when the calcium share with depolarization with the sodium and give the plateau potential depolarization. The gated calcium channel block is slow, require 10 to 20 times longer activation for the sodium. For these reasons are often called slow channel in contrast to sodium channel, which are called fast channels. Opening calcium provide more sustained depolarization, whereas sodium channel play key role in initiation action potential. So the sodium is the first depolarization, and then calcium, because it is the slow channel opening, and this is give plateau in action potential. The calcium channel is very important in cardiac muscle and smooth muscle. In this figure, we show that the depolarization by the calcium and give calcium muscle contraction in cardiac muscle and smooth muscles in the body. So in pharmacological science, the calcium channel blocker group of the drugs that block this channel and then reach to relaxation of the muscle. One of the subjects for the seminar is calcium channel blockers. Is very important in the treatment of cardiovascular disease. Plateau and some action potential. This is reached to. This is related to calcium ion in depolarization, as we show previously. This is a plateau depolarization related to calcium ion. transmission or propagation or conduction or conduction. Number one, many factors that affect on many factors that affecting on propagation or conduction or transmission. One of these factors is myelin. So when there is myelin in nerve, there is the propagation is very speed in the transmission of impulse. Myelin in central nervous system, brain and spinal cord by oligodendrocytes. But in peripheral nervous system is formed by the Schwann cell, as we show previously. This is lipid provide good in solution and preventing the movement of current across the cell membrane. Another factor that affect on the transmission or propagation or conduction is nodes of Ranvier. Nodes of Ranvier is without myelin sheath. So the action potential taking place in the nodes of Ranvier. This is give the jumping of the ions in this site and give the potency for the action potential. This is a phenomenon called saltatory conduction. Saltatory conduction is very important, significantly increase the velocity of conduction of the nerves impulse. So an unmyelinated axon is less velocity than that of the myelinated sheath of nerve. As we show in this figure, this is site of the Ranvier, nodes of Ranvier. This is action potential initiation, and then in the another point in P there is another jumping of ions, and this is given the velocity to the action potential and conductivity and transmission. This is jumping called saltatory function. Speed of impulse also affected by another factor such as the diameter of fiber and the temperature. So when the example for velocity of non-myelinated nerve 1 meter per second, but in the myelinated nerve 100 meter per second. This is a table as we show that when increase the fiber diameter of a neuron micrometer, increase the conduction velocity. As summary, in general, the greater in diameter of given nerve fiber, the greater its speed of conduction. The pain, one of the sign and for the action of the potential. This is another picture, and we need to remove this pain by local anesthesia. This is a group of the drugs is very important in medicine to prevent the pain. Another dosage form, one of these is the lidocaine, also used in medicine. This is the trademark carbol composition of lidocaine and epinephrine. So in the pain we reach to that to prevent the pain by using the local anesthesia. Local anesthesia mechanism of action of this group is another disease is multiple sclerosis caused by demyelination of a neuron, and so then there is many signs, symptom, weakness, numbness, and loss of bladder control and visual disturbances. All of this is related to demyelination, and when there is demyelination, this is of a neuron in the brain and spinal cord and optic nerve. The loss of myelin disturbed the normal conduction of the impulse. This is figure sign symptom of multiple sclerosis. The last in this lecture we take synapse. Synapse in this figure, all of this part called synapse. Synapse consists of presynaptic terminal and postsynaptic terminal, and this is synaptic cleft. How transmission the action potential, the impulse from the end on of the axon to the terminal dendrites of the cell of body of neurons by number one, calcium attached with these vessels contain a neurotransmitter, and then by exocytosis secretion the neurotransmitter in the synaptic cleft, and then attached to the receptors in post-synaptic membrane to transmission the action potential impulse from this nerve and to this nerve. The neurotransmitter is very important in transmission of the impulse. After end of its function neurotransmitter, the body are degradation of the neurotransmitter, such as acetylcholine by acetylcholine esterase enzyme to choline and acetyl-CoA. Neurotransmitter is very tight. Classification of neurotransmitters: number A, small molecules acting transmitter, class 1, acetylcholine, amine, adrenaline, epinephrine, noradrenaline, norepinephrine, dopamine, serotonin, histamine. Class 3, amino acids, alpha aminobutyric acid, glycine, glutamate, aspartate. Class 4, nitric oxide, NO. A neuropeptide, slowly acting transmitter, hypothalamic releasing hormone, pituitary peptide that act on gut and brain. As we show in this figure, cholinergic neuron, acetylcholine, acetyl-CoA plus choline, acetyl-CoA as the neurotransmitter by exocytosis receptor postsynaptic tissue, and then was this enzyme choline acetylcholine esterase enzyme degradation to choline and acetyl-CoA. And disease. The pH of the blood normally 7.4. When the alkalosis, the pH increase more than 7.4. This is increase the action potential, and the disease is called seizures. Yeah. One of these diseases is called Alzheimer. And at the end of the lecture, thank you very much.